this is basically where most of modern genetics, people in my lab, for example, do pretty much what I'm going to be talking about over the next few lectures. So this is now the meat of modern genetics. Okay? And so I'd like to take you through the methods first. Once we've finished, of course, with this lecture on gross chromosomal rearrangement, once we get into the small mutations, I'll first describe the different kinds of small deficiencies that can arise. And once I've described those kinds of deficiencies, then we'll go into a huge series of methods. And you're going to basically be applying those methods theoretically to answer genetic questions. This is basically what we do in my lab. Some of you have already worked in labs and might have done some of these techniques like small cloning experiments or PCR, things of that nature. Those are the methods that, and some of you are obviously interested in it because you guys have written to me wanting to work in my lab. We'll talk about this. Okay? And this is really the place where genetics is right. And we're still going to go historically through. So we'll start with methods that were developed in the 80s, 70s, and 80s, and then gradually move into the very recent methods, which are all genomic-based. We analyze all of the genes of the person. So we gradually move into. Okay, so we were talking about inversions, and I believe we finished talking about inversions. Segments of DNA or chromosomes will invert. When they invert, there is no loss or gain of genetic material at the time when the inversion takes place, right? It's just a flipping of the DNA. However, once that individual goes through meiosis and there is a crossover in the region that is inverted, then you have problems. All of the different types of gametes that are produced, if from recombination events that have taken place within the inverted segment, will be inactive gametes. And those gametes will be non-functional and they will be lost. All right? So it's only when you have a crossover or a recombination event within the inverted segment. If it happens outside the inverted segment, because there's no loss of genetic material, because the inverted segment still has all of the genes. Now let's look at another gross chromosomal rearrangement, and that is, okay, translocations. Oh, we talked about this. Translocation. So translocation basically is where one segment of a chromosome hops onto another chromosome, basically. All right? So you can either have a reciprocal exchange of segments from two different chromosomes, or you can have one piece of a chromosome just jumping onto another chromosome on its own without any reciprocal event. And that's known as a translocation. So we're going to talk about translocations where there are reciprocal events, just to keep it simple. Here is our hypothetical chromosome. A, B, C, D, E, and F are the bands on this chromosome. Here are two different homologous copies of this chromosome. And then there is another chromosome with bands O, P, Q, R, S, and T. Okay, so we have one chromosome here and we have one chromosome here. This is the normal order of the bands in a normal wild-type individual. Now you have a reciprocal translocation, where a segment of this chromosome will hop onto this chromosome and a segment of this chromosome will hop onto the other chromosome. So you're exchanging arms, or pieces of an arm. And so here's what happens. You go from an A, B, C, D, E, F to an A, B, C, D, S, T. This segment of this chromosome has jumped onto this chromosome. Simultaneously, this segment of the chromosome, the E and the F segments, have jumped onto this chromosome. Now it's only happening on one of the two copies, obviously, not both copies of the chromosomes. I'm just showing you those one copy where the translocation has taken place. And this is a reciprocal translocation. And so, very similar to an inversion, you don't lose or gain any genetic material at this point. When you've gone through a translocation, You haven't lost any genes, you haven't gained any genes, you haven't lost any piece of DNA or gained any piece of DNA. Usually. Question? We'll come to that in a minute. That's a very good question. What happens with crossing over when you have something like this? And we're coming to that. And that's where you start to have problems, as you will see in a minute. So it's usually with these kinds of gross changes or inversions and translocations, if you are not going to go through meiosis, you are more or less normal. It's only when you go through meiosis, your progeny are the ones that are going to have problems. If there is a crossover in the region that has been inverted or translocated, and those are the gametes that get fertilized. So there's a very rare probability of this happening, but in a population of 5 billion people or trillions of little organisms, you will see effects when you look closely.
Is this happening between homologous chromosomes or like different chromosomes?
We'll come to that. For the translocations, we'll come to that. For the inversions, you saw they were happening within the homologous chromosomes. Translocations also are homologous chromosomes, but they are 4 chromosomes. Both pairs of these chromosomes are going to be. So we'll see. All right. Any other questions? All right. So here is a balanced translocation between chromosome 21 and chromosome 7. Right. So this segment is actually a segment of chromosome 7, and this segment is a segment of chromosome 21 that have fused together. Here is the other version of it, 21 and 7. The 7 chromosomes split and this 21 chromosome split, and you join the 2 halves together. This is normal 21, this is normal 7. This is one translocated 721, this is the other translocated 21 7. So you see how it is, it's a balanced translocation. So you basically, the long arm of chromosome 7 and 21 broke off and switched places, basically. So that's what you basically end up with. And here is an unbalanced translocation where you have normal 7 chromosomes, but you have a segment of a 7 onto a 21. This is known as an unbalanced. Here, you have actually gained additional genetic information. And I'll come to how you gain extra genetic information in an unbalanced translocation. This doesn't happen at the very first stage. It happens when you go through meiosis. And so we'll see why and how this happens. The nomenclature for translocations are very similar to what you've been getting used to. You usually denote a translocation with the letter T. And you basically then indicate which 2 chromosomes have translocated. So it's chromosome 11 and 18, for example. And then which segments have translocated. For example, Q21 and Q21 have translocated. In this example, this is chromosome 9 and 22 have translocated, and it's segment Q34 of chromosome 9 and Q11 of chromosome 22 that have translocated. So you denote it in simply the fashion and the chromosomes that have translocated. Of course, you also have additional information. You indicate how many chromosomes there are. You indicate whether the person is a male or a female, like we did earlier. So you would say 46 XX, and then P11, 18, Q21, Q21, or something to that. All right, so what happens now? Let's imagine a person has undergone a translocation in their germ cells. Okay? Now the germ cells are going to go through meiosis. What happens when they go through meiosis? Right, so here is one chromosome, A, B, C, D, E, F. That's the normal chromosome. And then here is the translocated chromosome, A, B, C, D, S, T. And then you have the second chromosome, O, P, Q, R, S, T. That's normal. And then the reciprocal translocated fragment, O, P, Q, R, E, F. So this E, F from this chromosome jumped here, and the S, T from here switched here. Okay, so now how do you go through meiosis when you do this? In meiosis, homologous chromosomes pair, right? Well, what's going to happen here? How do you pair with these weird chromosomes? So, The key to notice is that the individual has a normal amount of genetic material. Correct? There's no gain or loss of genetic material here. Right? And so every gene is still present in 2 copies, or every band in this instance. So, but it's rearranged. And so, when you basically end up in this fashion, let's see how they pair. How will the translocated chromosomes pair in meiosis? That's the question we're going to look at. And they pair basically across both chromosomes. The segments that are present will pair with the segments that are present, irrespective whether there are 2 chromosomes or 4 chromosomes. And they pair, and you can actually see the paired chromosomes under the electron micrograph or under a light micrograph as basically a cross-shaped structure. And this is a picture of this. This is a chromosome translocation in maize that I'm showing you. You can see very clearly that there's been 4 paired chromosomes. They're forming a cross. And if you look at it in this fashion, here are the 4 chromosomes that we were talking about. So here is the normal chromosome, A, B, C, centromere D, E, and F. This chromosome is the normal one. Let's call it N1. And then you have the translocated chromosome. A will pair with A, B will pair with B, C with C, and D with D. But then you diverge away because S can't pair with E and T can't pair with F anymore. So they basically form a tail initially, right? Let's pretend we're starting to go through a pairing event. And I'm going to just pretend we're going through a translocation or pairing event. So we have chromosome A. This is N1. And then this is the segment of N2 that's pairs nicely. But then the pairs don't pair anymore, so they stay flapping around, right? Similarly, you have the second chromosome, the second normal chromosome, that goes like this. And then it pairs with its segment, and then the segment pairs like this. And so you get complete pairing in a sense. And so that's what you see here, that N1 pairs with part of the translocated chromosome, that other segment hangs out, then the part of the other translocated chromosome will pair, and then this will pair in this. So now you have paired chromosomes, right? And then you go through meiosis. Okay, now comes the problem. You're going to go through meiosis, right? What happens in meiosis? You pair, and then the spindle attaches to the centromeres. And usually you have 2 chromosomes, and the spindle attaches from either side, and it pulls the homologous chromosomes to either end, right? In meiosis 1. Well, here you have 4 centromeres. And the cell doesn't know which centromere to interact with. The spindle has no idea which centromere to interact with. It's just looking around, finds a centromere, it attaches to it. So the spindle will attach to these 4 centromeres entirely at random. You have 2 spindles, you have 4 centromeres, so 2 of the ends will attach to one set of one spindle pole will attach to 2 of the centromeres, the other spindle pole will attach to the other 2 centromeres. But that attachment is entirely at random. So imagine you have a spindle here, and you have a spindle here. Okay. You have one spindle here and one spindle here. This spindle here will attach to this centromere and this centromere. The spindle over here will attach to this centromere and this centromere. That's one possibility. So, let's draw the centromeres. And let's imagine there is a spindle here and a spindle here. So this spindle will attach like this. This spindle will attach like this. And it will pull the 2 chromosomes apart. Correct? When you do that, one pole let's label this. This is your T1, and this is your, this is N2. This is T2. Okay. So now when you pull these chromosomes apart, you're going to get in meiosis 1 an N1, T1, and an N2, T2 homologous chromosome in the gamete. Correct? Yes? So, when you look at the genes that have gone along with this, you're going to get N1 and T1, so you're going to get A, B, C, D, E, F, correct? And you're going to get O, P, Q, R, E, F. So you've duplicated the EF gene, and you've lost the ST gene in one of the gametes. In this gamete here, you basically duplicated the EF gene, but you lost the S and the T genes. In this gamete here, you will basically have A, B, C, D, S, T, and O, P, Q, R, S, T. You will duplicate the ST segments, and you will have lost the EF segments. So your gametes will not be normal. You will have lost genetic material or gained genetic material due to the translocation once you go through meiosis. Correct? That's in one orientation where the spindle is here and here. But the cell doesn't know how these chromosomes line up. Imagine you have the spindle here and here. Right? That's also possible. Chromosomes are three-dimensional entities, correct? So you can imagine a cross standing up like this or like this. Varies. The spindles are fixed. Let's imagine these are the two spindles there. And I am a cross. I can be like this, or I can be like this, or I can lie down on the floor and be another cross. So I have three different ways in which I can line up at the spindle. And so the spindle is going to attach. So if it attaches on, if the chromosomes line up in this fashion, then this centromere and this centromere will go to this spindle. This centromere and this centromere will go to this spindle. And now you're going to end up with chromosomes N1 and T2 here, and N2 and T1 here. Right? Is that clear? So that's one other possibility. But once again, when you analyze these segregated chromosomes, you will see that you've either gained or lost genetic material. If you have the poles here and here, let's imagine you have the pole here, you will get a chromosome that has OPQREF and OPQRST. So basically, you're going to have, you notice what happens here. You've got a huge duplication of the O, P, Q, and R genes, and you don't have any of the A, B, C, D genes at all. Right. So once again, you'll get unbalanced gametes. Okay? Okay, that's two orientations. Go ahead.
So in or non-germ cells, transcription and translation occur normally?
Yes, usually. You'll see examples where they don't, but usually no problems when you have a translocation. Rare instances, you have problems, and those rare instances are actually life-threatening when they do happen. And I'll give you examples of that. But that's in mitosis and in normal body cells we're talking about. Very good question. We'll come to that in a minute as well. But for now, okay, so you're going through meiosis, you have problems. Now there are two orientations I talked about. A spindle here and here, and a spindle here and here. But there is a third possibility of a spindle attaching. And that third possibility is that the spindle is in front of this picture, and the spindle is behind the wall. Okay. So you can have a third possibility of the spindle. And now you might imagine that N1 and N2 go to that spindle, T1 and T2 go to the other spindle. So you are attaching and pulling them up like this. So one spindle attaches to N1, the other spindle attaches to N2 and pulls it out of the wall, and then T1 and T2 go through the wall. And so the third possibility is you end up with chromosomes that are normal because you have N1, N2, and T1, T2. Those are the chromosomes that look like the parent, and they have no loss of genetic information. So 1/3 of the meiotic products of a translocation are normal, 2/3 of them are abnormal. Now, this is in theory. In practice, the numbers are slightly different because these chromosomes do have a preference of how they line up at the single pole. And because of that, the ratios of these three different possibilities are not exactly 1/3, 1/3. But theoretically, you have three different ways in which you produce the gametes, and only one out of those three ways you will get normal gametes. The other two out of the three ways, you get totally abnormal gametes. You lost a huge amount of genetic material, or you duplicated a huge amount of genetic material. And once you get that weird genetic material, you have what's called an unbalanced translocation. That's the picture I was showing you earlier, an unbalanced material. This one, an unbalanced translocation. This is an individual that has gone through meiosis, basically, and generated something.